This is interview number 6.
WARN, of course, stands for the Worldwide Antimalarial Resistance Network. It was set up about 6 or 7 years ago with the aim of generating intelligence, high-quality evidence to inform policy on malaria treatments around the world. So to do that, we collect information from researchers around the world who are working on testing antimalarial medicines in the field, and to try and generate evidence to say, well, in this particular location, this is the best treatment right now, and then to keep doing that over time, because obviously, you know, malaria, the situation in terms of resistance and efficacy is dynamic. You have to keep monitoring it. So we generate, rather collate all that information. So historically, resistance has somehow always come out of Southeast Asia. There have been other foci where resistance has developed in South America. But typically what happens is that resistance will develop in Southeast Asia and it will be exported across India and into East Africa and then the rest of Africa. And that's precisely where the problem is. It is just that if the resistance that is developing here right now does get exported into Africa, the impact in terms of morbidity and mortality is much higher. So the problem is getting worse because we're now starting to see ACTs to some extent failing. This is related to the fact that artemisinins, the parasites in the region, have become tolerant, which means that the partner drugs are more exposed to the parasites, and that has led to partner drug resistance evolving, changing over time. And yeah, we need to track that. So that's what we're doing through the Track 1 and Track 2 studies.
Why does this line of work matter, and why should we fund it?
Well, it matters because malaria is a disease, as I mentioned before, that can have devastating consequences on the lives of people. It's difficult to prove a causal relationship, but you know, wherever there's malaria, there's poverty. Whether one causes the other, it's really difficult to tell. But you know, we want to make sure that people are not suffering from a curable disease all the time, which affects their performance at school, their performance at work, you know, their economic viability and that of the country. So it is important to make sure that people who are living in malaria endemic regions get the best possible treatment. And one way of doing that is to have a well-developed, robust system for surveillance of resistance, which will help make sure that, you know, people get the right treatment at the right time. The other thing that we of course realize is that something that plays into resistance is poor quality medicines. So you do get cases where there are medicines that do not contain enough of the active ingredient, and obviously if you're giving a suboptimal dose, that does you know, promote the development of resistance. So there is the drug quality, but there's also pharmacokinetics and pharmacometrics. There's the in vitro module, there's the molecular module, there's the kinetic module. All of those guys, they try and collect different pieces of evidence from all over the world, wherever these kinds of studies are done, to come up with recommendations on treatment and dosing.
What important lines of research have emerged in the past few years in your field?
I think what has become increasingly important is to have as broad a network for surveillance of resistance as possible. So, I mean, there was a study which was led by Dr. Chamyo Tun, who is also working with us in the unit, and you know, they collected samples from all over Myanmar, and it was possible to map the extent of the spread of, or independent emergence, if you will, of artemisinin resistance.
How does your work fit into translational medicine within the department?
The mapping of resistance is a good example. The samples that we collected through the TRACK study actually helped with the clinical validation of the K13 marker, which is a genetic marker of resistance to artemisinins. Now we're trying to kind of roll it out and, you know, try and get as broad a coverage as possible in terms of the prevalence of the resistance marker. And that is, you know, we're translating genomics into something that is a relatively easily implementable surveillance tool. So I would say that that's a good example of translational medicine. What I enjoy is that knowing that I'm working, doing something that is useful. I think it's as simple as that. I mean, I have some background in working in a biotech company before, and it was just not satisfying. But working here at Moru and before that with MSF in Africa, I mean, the feeling that I knew that I was doing something useful was deeply satisfying, and I think that's one of the best things ever.